Hello, my name is Anaya Scott, and this is my gut microbiome presentation. Here is an overview of what I'll be discussing in this presentation. I'll be discussing things like the importance of a microbiome, the comparisons of our samples, and what we would do in future experiments. Please feel free to pause and read. Here is my introduction. To start, what is a microbiome? A microbiome is a community of microorganisms living in an environment. But what about a gut microbiome? A gut microbiome is the collection of microorganisms present inside the gut of an animal. Microbiomes are important because they impact the health of humans and other animals, and it correlates to diseases and or disorders such as IBS, autism, and gastric cancer. As I mentioned previously, IBS correlates to gut microbiome health of humans and animals. But what exactly is IBS? IBS, or Crohn's disease, is a disorder that affects the large intestine. A study done by Matthew Bull and Nigel Plummer found that humans with healthy gut microbiomes have direct bactericidal effects, preventing bacteria from adhering to the intestinal wall, making them less likely to have IBS. Those with IBS have an alteration in their microbiota composition, disturbing fermentation in the colon, playing a major role in IBS symptoms, making their relationship correlative. Metabolic diseases, such as obesity, was first experimented on mice. Experimenters found that mice with the mutation in the leptin gene was shown to have different gut microbiome than the mice without the mutation. Lean mice with the healthy gut microbiomes were heavily influenced by the phylum Bacteroidetes, which have been shown to positively correlate to body fat percentage. Obese mice with the mutation in the leptin gene were found to be impacted heavily by firmicutes, which have been proven to be negatively proportional to resting energy. In humans, varying microbial compositions causes different variations in the way people harvest energy, which explains and is responsible for diseases like obesity and diabetes, making the relationship to gut microbiomes correlative. Every hypothesis must come from a starting question. Our question was, will age have an effect on the gut microbiome? Based off of our question, we determined that if we take fecal samples from dogs with different ages, then there will be a difference in the gut microbiome of the younger dogs compared to the older dogs. Here are our cited resources. Now on to methods and materials used. Here are the dogs we are sampling for this specific experiment. So for each dog for age, we kept it as a 2-year gap, seeing as our youngest dog is 4 years old, our second youngest is 6 years old, our first oldest is 10 years old, and the oldest dog we are testing today is 12 years old. Our younger dogs are both female, while our older dogs are both male. All of our dogs have relatively good health, 3 of our dogs being very good, in that category, and one of our dogs being average. Our Chihuahua gets occasional exercise as well as our Chow mix lab, but our youngest and oldest dog both do not get exercise. When conducting our experiment, we followed the instructions listed per our lab manual, which I've cited below. Starting with the purification of the DNA, this was done using a QIAGEN kit. Then we homogenized the stool samples in a buffer in order to prevent the degradation of the DNA. We then bound the DNA to the filter in the spin columns, and then after that we rinsed out the contaminants such as proteins and RNA before releasing the DNA into the tube using the elution step. In order to see how much DNA we had, we used the UV spectrophotometry to determine the concentration of DNA in our samples. We used the result of our 260/280 ratio to determine the purity of our samples. As long as it was between the range 1.5 and 2.2, our samples were considered pure. Otherwise, they were contaminated. So to do the qPCR, we used two primers, the forward primer and the reverse primer. One primer tested for the kingdoms, such as bacteria and fungi, while the other tested for phylums, which we picked Acetobacteria and Firmicutes. The SYBR is an asymmetrical cyanide dye used to stain the DNA so we can quantify the amount of double-stranded DNA present in our qPCR run. We included two control groups, positive and negative. Our negative control group replaced purified DNA with water, which was used to test for cross-contamination, and our positive control group we used pure DNA from E. coli, which would help us identify if there was a problem in our isolated DNA samples. Here's our results. Based on our spectrophotometry results, our ratios fell well within the range of our protein and RNA ratios, and all of our DNA samples were deemed pure. In our kingdoms, we had no results in the positive controls, but we had some contaminants in our negative controls. For each sample, there was a good amount of U bacteria present. However, fungi was either rare or absent from our samples. When testing our phylums, we found that our negative control groups contain contamination, and it is over 25, so our contaminants are considered rare. The positive control group is a bit higher than expected due to possible incorrect technique when micropipetting, DNA contamination, or the PCR reaction not working properly. Our first sample has some Acetobacteria and Firmicutes. Our second sample shows that Acetobacteria is rare or absent, but there are Firmicutes present. No Acetobacteria was found in samples 3 and 4, but there is evidence of Firmicutes in both. Based on our anti-log results, age is not a discriminatory factor for kingdoms, but it may be for the phylums. When examining the results for the phylums, we discovered that Firmicutes and Acidobacteria were rare or absent. In younger dogs, in older dogs, Acidobacteria is absent and Firmicutes are either abundant or rare, signifying that as the dog ages, there may be more Firmicutes because the 12-year-old dog had more than our 10-year-old dog. Lastly, here's our discussion section. Based off our results from the spectrophotometer, our DNA concentration for sample 1 was 45.7 nanograms per microliters. For sample 2, it was 139.7 nanograms per microliters. For sample 3, it was 31.9 nanograms per microliters. And for sample 4, it was 13.5 nanograms per microliter. Based off the instructions from the Qiagen manufacturer's site, we did not have enough DNA since we need about 250 to 500 nanograms of DNA to actively run a successful qPCR. But in terms of quality, all of our samples were pure since they fell between the ranges of 1.5 and 2.2. In our first qPCR run, there wasn't any bacteria found in our positive control groups. However, in our negative control groups, there were bacteria present, but it was considered rare at the value of 25.39. And in our second qPCR run, the negative control group was effectively absent with the value of 32.09. Based off the results of our samples, for our 4-year-old Frenchie, the bacteria is rare. For the 6-year-old Chihuahua, there is abundant bacteria. For the 10-year-old Chow mix, there was rare bacteria. And for the 12-year-old Beagle, there was abundant bacteria. However, it seemed that fungi was absent in all dogs we've tested. For our phylum group, for our Frenchie, the Acidobacteria is absent and the Firmicutes are rare. For the Chihuahua, the Acidobacteria and Firmicutes were both rare. For our 10-year-old Chow, the Acidobacteria is absent. However, the Firmicutes are rare. And for the Beagle, our Acidobacteria is absent. However, the Firmicutes are abundant. So, was our hypothesis supported or rejected? Our hypothesis was supported as there was a difference in the gut microbiomes of the dogs we've sampled. However, in the future, we would test more samples with the same age gap, but using the same gender instead of varying between male and female to determine if the microbiomes are different with just age as a factor instead of age and gender.